like to introduce Catherine Wagner, Katie. Katie is an assistant professor in the Department of Biology and Diversity Institute at the University of Wyoming. She is our fish person, and I say that in the best way. Katie will talk to us about the evolution and diversity of the East African cichlid fish. And I don't want to take up too much time. I have a whole thing I was going to say about you, but I'm going to actually have you come up and give your talk since we're running very long.
Okay, it's wonderful to be here today, and I am glad that I can take the focus a little bit away from humans and move towards fish. So just to introduce you a little bit to these fish called cichlids. This is a family of fish. They're diverse in South America and in Central America. They're also found in southern India and in the Middle East. And even if you don't think that you are familiar with cichlids, you may actually be familiar with them on your plate because tilapia is an African cichlid. And tilapia is one of the most commonly farmed fish in the world. So tilapia is one of many thousands of cichlids that live in Africa. They're exceptionally diverse. And within the East African Rift Lakes alone, there are more than 1,500 endemic species of cichlid fishes. In the three Great Lakes, Victoria, Tanganyika, and Malawi, there is the independent evolution of hundreds of these cichlid species. In each of these lakes, these fish have exceptional diversity in ecology and in morphology. And they form independently in each of these lakes what we call adaptive radiations, where adaptive radiations are the rapid origin of ecological and species diversity via descent with modification from a common ancestor. So this process of adaptive radiation has happened independently in each of these three Great Lakes and also in many other lakes across the African continent. So in this process of adaptive radiation, from a generalized ancestor, we have the evolution of specialized ecology such that within each of these cichlid adaptive radiations, we have the evolution of fish that are specialized to eat scales of other fish, fish that are specialized to eat algae off of rocks in all kinds of different ways, fish that have funny fat lips that help them eat invertebrates between cracks in rocks, other fish that have these jaws in their throat called pharyngeal jaws that help them crush mollusks, and also all kinds of specializations to eat other fish. So these are what we call piscivores. You can also see that these fish are exceptionally diverse in color and in body shape and size. And the thing that I want to emphasize here is that within these cichlid adaptive radiations, we're essentially evolving entire food webs. One thing I like to say is that the only thing that cichlids can't do is photosynthesize. So we're evolving everything from the primary consumers in these ecosystems up to the top predators in these ecosystems. And this is, as I mentioned before, happened in dozens of lakes across the African continent. And this plot in the red shows lakes where there has been the evolution of endemic cichlid species. And in gray shows lakes where there are cichlid species, but no endemic species have evolved there. So if we were to draw an analog with another famous example of adaptive radiation, these are Darwin's finches from the Galapagos. And Darwin's finches are famous because from a single common ancestor in the Galapagos, there has evolved about 18 species that have diverse bill morphologies that allow them to eat all sorts of different things. If we were to draw an analogy between Darwin's finches and cichlids, this would be as if Darwin's finches had done this not only once, but dozens of times, and if some of these Darwin's finch radiations had hundreds of species instead of the 18 species that currently exist there. So this lends itself to thinking about African cichlids as a grand natural experiment, where we can ask questions like, why do adaptive radiations happen sometimes and not others? And what determines the number of species that evolve within adaptive radiations? So I'm going to touch on three questions today that are related to research that I've been working on. The first is what predicts when adaptive radiations happen. The second is what influences the number of species that evolve within these lakes. And then third, what enables exceptionally rapid diversification in some cases. So if we look at the diversity of cichlids across the African continent, we can identify 38 times where cichlids have entered a lake and have subsequently diversified. But we can identify another 145 times where a species has entered a lake and has failed to diversify. So this data sets us up to ask the question, why do some lineages diversify while others don't? So to address this question, we put together a data set including traits of these species that were hypothesized to influence speciation, the origin of new species within cichlids. So these are intrinsic factors related to the biology of the organisms. We also included a lot of potential predictors from the environment, aspects of the lakes themselves, the environment that these fish encounter when they colonize a lake. And so these are extrinsic factors. And when we analyze all of these potential predictors and models together, we invariably find that the best models include both intrinsic and extrinsic factors in predicting when cichlid adaptive radiations happen. And so this leads us to a picture of thinking about cichlid adaptive radiations as happening as an interaction between the intrinsic biology of the organisms and the environment that they encounter. Specifically, 3 of the predictors that come out as strong positive influence on cichlid adaptive radiation are the depth of the lake that the cichlids are found in, energy, so this is solar radiation hitting the lake, and also the presence of sexual dichromatism, which is differences in colors between males and females, and that strength of sexual dichromatism is often related to the strength of sexual selection that operates in these cichlid lineages. And in particular, we were interested to find both depth and sexual dichromatism as predictors of adaptive radiation, because both of those aspects are involved in one of the best studied mechanisms of cichlid speciation, which is known as sensory drive speciation. This is where there's differential adaptation along depth gradients in lakes, where at deeper depths in a lake, the fish are living in a very different light environment than the fish have at the shallow part of the lake. And this drives differential ecological adaptation and also differential sexual selection, which leads to speciation. So finding this pattern of both depth and sexual dichromatism at a broader scale for cichlids suggests that this mechanism may operate across the continent and help us to explain cichlid diversity more generally. We were also interested to find energy coming out as a predictor of cichlid adaptive radiation. And although this hadn't ever been discussed in cichlid biology before, we know that energy availability would have a strong influence on resource environment and on population abundances, and both of those aspects we would expect to play into the evolutionary biology of these fish. So this presents lots more questions to ask about the relationship between energy availability and evolution. And one thing I want to mention before moving on is that these models are only as good as the predictors that we can include in them. And there are certainly other predictors which are also really important in explaining cichlid adaptive radiation. One of those that is emerging as incredibly important is genetic diversity of these lineages and their history of hybridization. And that's a subject that I'll speak about towards the end of the talk. I also want to point out that surface area of lakes does not predict whether or not cichlid adaptive radiations happen, but surface area becomes very important when thinking about predicting the number of species that evolve within these adaptive radiations. So that's the second part of the talk, asking the question, what influences the number of cichlid species that evolve in these African lakes? So in this plot of Africa, you can see the red dots correspond to lakes where there has been evolution of new species. The black dots are lakes where there has not been the evolution of new species. And the size of the dot corresponds with the number of species in that lake. And you can see that these lakes range in species number from many hundreds of species down to just a few species. So we can think of the number of species in these lakes in a couple of different ways. Some lakes, the species are there simply because they've colonized the lake. They've dispersed into the lake from rivers. And so this would be a dispersal assembled community. We have other communities where there's been evolution of one of those colonists. And so we can think of this as an evolution assembled community. And in these cichlids, sometimes this evolution has happened in one lineage, and sometimes it's happened in multiple lineage to contribute to the number of species that we find in that lake. If we plot the number of lineages we find in a lake, so this is the number of species essentially as a function of lake area, and this is not accounting for the effects of evolution. So this is cutting out all the species that have evolved in place. We see that there's a positive relationship here. So we get more species in the lake simply the bigger the lake is. More species colonize into it. But if we then add on the effects of evolution, what we see is that evolution within these lakes produces, on average, an order of magnitude more species in these lakes than dispersal alone. So evolution is having a gigantic impact on the number of species within these lakes. And additionally, more species evolve in the very largest lakes. So we can predict the number of species that evolve in a lake based on the size of that lake. If we take this one step further and ask, how does the rate at which species evolve influence these patterns? This is a plot of diversification rate as a function of lake area. And what you can see here is that diversification rate does not predict the number of species in these lakes. So there's no relationship between diversification rate and lake size. There's one clear outlier in this plot, and that is Lake Victoria. And if we look at the plot of species richness in lake area and look at where Lake Victoria is on that plot, we can see that Lake Victoria's cichlid diversity is exceptional because it both has an exceptionally rapid diversification rate and because the lake is very large. And so that points us to Lake Victoria as an outlier and begs the question, what enables this exceptionally rapid diversification in the case of Lake Victoria? And so this question about the exceptional rate of evolution in Lake Victoria is actually an old one in evolutionary biology. And Lake Victoria is less than 15,000 years old. It was dry 15,000 years ago, and it has about 450 cichlid species. And this exceptional rate of evolution has led many people to suggest that perhaps these aren't actually species, perhaps it's just a hypervariable population, and we shouldn't really consider these species. We used genomic data about 5 years ago for the first time to have a really in-depth look at these species genetically, and we find that when we use enough genomic data, we can very readily tell these species apart. As you can see, each of the species in this plot forms nice groups when we use enough genomic data. So we really think that these are species as we would consider anything a species. And thanks to collaboration with the Broad Institute, we now have 5 fully sequenced and annotated reference genomes that span a lot of diversity in African cichlids. They're from many parts of the African cichlid evolutionary tree. So using these resources from the annotated reference genomes, we can dig further into understanding what are the genetic differences between these young species and what are the functional consequences of those genetic differences. This relates to we've been using this genomic information to ask these questions about the origins of exceptionally rapid species diversity in Lake Victoria, specifically asking the question, where did this Lake Victoria radiation come from? And what genomic properties facilitate this extremely rapid radiation? And the work that I'm going to talk about now is led by Joanna Meyer, who is now a postdoctoral fellow at Cambridge. So Lake Victoria is part of a larger group of cichlids called the Lake Victoria Region Superflock. This includes cichlid adaptive radiations in Lake Albert, Lake Edward, Lake Kivu, and also Lake Victoria. So all of these radiations together form this group called the LVRS, the Lake Victoria Region Superflock. And this comprises about 700 species that have evolved in the past 150,000 years. So the origins of this group coincide roughly with when modern humans were leaving Africa, to connect back to our earlier talks. So when we look at the genomic data from the Lake Victoria Region Superflock, using methods from human genetics, we infer that there was a hybridization event between two divergent lineages, which subsequently led to the Lake Victoria Region Superflock. And that hybridization event was between the Congolese lineage, whose distribution is shown in red on the map here, and an Upper Nile lineage, whose distribution is shown in this map in blue. So these two lineages were at least 1 million years divergent from each other. They encountered each other in the Lake Victoria region, and subsequently, all of these adaptive radiations in these different lakes in the Lake Victoria region emerged. So the question then becomes, was this just a happenstance event in their history, or did it have functional consequences? Is there a functional consequence to this admixture in the same way that we can ask questions about the functional consequences of Neanderthal admixture in human history? And so one piece of evidence pointing to functional significance of this hybridization event comes from the LWS opsin gene. LWS opsin is long wavelength sensitive opsin. So this gene is involved in visual acuity in different light environments, where one variant of the gene, so this is the gene's diversity in cichlids from the Lake Victoria region superflock. And we find in some species a version of the gene which gives visual acuity in clear water over rocky substrates. And in other species in the Lake Victoria region superflock, a version of the gene which provides visual acuity in murky water and muddy substrates. And so these different versions of the LWS opsin gene have been repeatedly used for adaptation to these different light environments. When we look at the versions of the gene present in the Congolese lineage and in the Upper Nile lineage, we see that all of the Congolese lineage species have the clear water variant of the gene, and all of the Upper Nile lineage fish have the muddy water variant of the gene. And so this suggests that the reason that we find these two variants of the gene together in the Lake Victoria region superflock is because of this history of admixture between the lineages. So this lends evidence to the idea that this ancient hybridization event provided functional genetic variation, which was subsequently used for adaptation within these adaptive radiations and facilitated the rapidity at which these radiations could happen. So in summary, we think that hybridization facilitates cichlid evolution by providing functional genetic variation and enabling extremely rapid adaptive radiation. And we also think that these kinds of hybridization events may be quite pervasive in cichlid evolutionary history and help to explain rapid adaptation and rapid adaptive radiation many times over. So in summary, the take-home points from my talk are, first of all, that we need to consider interactions between intrinsic factors and extrinsic factors in predicting when adaptive radiations happen. Second, that lake area predicts the number of species that evolve in African lakes, and that that's somewhat disconnected from the diversification rates in these lakes. And third, that hybridization may be key in understanding the exceptionally rapid adaptive radiations in African cichlids. And with that, I'd like to thank my collaborators and funders, and I'd be happy to take some questions.
Thank you, Katie. We have a question?
What about us like a cichlid, a cichlid compared to other types of species? It makes it so good at like diversifying and like adapting.
Yeah, that's a fantastic question, and it's why so many of us keep on studying cichlids, because they're so exceptional. If you look at other fish groups in these lakes, some of them have evolved a few species here and there, but nothing like cichlids, nothing like the hundreds of species that we see in these lakes. I don't think there's one answer. I think that there are a number of features of cichlids which facilitate their rapid evolution, but we would like to think that some of these things that I talked about, propensity towards sexual selection, propensity of hybridization events, and sharing of these genetic variants across divergent groups are some of the reasons why they evolve so rapidly and so many species.
Thanks very much.
Heather Ramoff, I'm wondering how the species, the factors that maintain species integrity, are they largely behavioral or genetic?
Many of the features that maintain species integrity are behavioral. So they're what we would call prezygotic. They're mate choice decisions. And so that's how hybridization can happen very readily. That's not to say that those behavioral differences don't have a genetic basis also. Yeah.
Alyssa?
Hi, I'm Alyssa Goodman from here at Radcliffe, and I was really admiring your beautiful images of all the fish, and it made me wonder whether or not there's a good correlation between the genetic differences and the actual appearance of the fish. Like, do the fish know that each other are different species, and can they even see the colors of the fish?
Yeah, that's a great question, and I wish it was easier to ask every one of those species this question. So the mate choice decisions, as I just said, are a really crucial thing for maintaining species boundaries in many, many cases. And so we know, at least in those cases, that they can tell each other apart, and that's very important for the maintenance of diversity. Yeah.
Yes?
I was just wondering what is known, this is Tim Havel from MIT, just wondering what is known about horizontal gene transfer in Cichlids.
Yeah, so in the case of sexually reproducing organisms like this, we think of hybridization as horizontal gene transfer. That's our analog. Yeah. Yeah.
We have 2 more minutes. I have a question. So you mentioned the energy impacts the diversity. Is that the same as temperature?
It's not temperature. So the proxy that we used was solar radiation to these lakes, which we would think would have a direct impact on productivity, so primary productivity in these systems. It would be fascinating to have a global or a continent-wide data set on productivity instead of just using solar radiation as a proxy, because there's all kinds of complex limnology that translates solar radiation into the resource base for these lakes.
Is there information on temperature?
There's not. We don't have information on temperature in our system. Again, that's another thing that would be great to have for all these lakes.
Right. Well, that was fantastic. And now we have our last speaker. Thank you, Katie.